Thank you, Sarah. Good afternoon, ladies and gentlemen, and thank you very much to the organising committee for inviting me to come and speak to you today. So this is the last lecture in this session on targeting breast cancer care with MRI, and it focuses on a novel and topical area of imaging that some of you may have heard of, particularly if you've been to some of the larger imaging or radiology conferences recently. But equally, it might be a term that's relatively new to many of you. It's called radiogenomics, and so over the next 20 minutes or so, I'm going to give you an introduction to what radiogenomics is, where we are with it, and how we might be able to use some of these techniques towards a more personalised treatment programme in the future. So we'll look at what radiogenomics is, we'll look at some definitions, and we'll look at where our data pool is coming from for this sort of analysis. We'll talk about why radiogenomics is important in breast cancer and how we might be able to use these techniques in the future. We'll look at some historical data, where we are now and where we might be going in the future. So radiogenomics refers to the extraction and analysis of imaging characteristics from different imaging modalities, linking that with the genetics, or in the case of breast cancer, in the most simplistic level, to the molecular subtype of breast cancer, in order to determine specific imaging phenotypes or imaging traits that have distinct patterns and may in themselves have significant predictive or prognostic value. So the aim is to capture standard descriptive and quantitative imaging phenotypic differences automatically using advanced bioinformatics techniques. And the aim is to determine specific radiogenomic signatures or imaging phenotypes or imaging traits that may in themselves be able to give information on long-term outcomes, so the likelihood of metastatic disease or recurrent disease. To date, the vast majority of this analysis has been done on MR data, specifically three-dimensional multiparametric MR data. And the thinking behind it is that this kind of quantitative data may provide more complex information on lesions that would otherwise be missed. So, for example, we know that when we report our breast MRI studies, we give a morphological description of the abnormality. As you can see, the cancer we've got in that image here, we've drawn a region of interest around it. The colour map within it shows that there's quite a lot of heterogeneity. Now, depending on what MR sequences you've done and what software packages you have available to you, you can choose a number of parameters. Here we've got the initial area under the gadolinium curve. For every single voxel within that region of interest, you can get a value for that specific parameter. And you can see in the image on the right, we've plotted that as a histogram. We can then go and sum that for every single slice containing tumour to get representative whole tumour quantitative data sets. This is otherwise known as big data sets. So depending on the size of your tumour, you can have any number of parameters for one cancer, anywhere from say 50, 500, 2,000, or even up to 10 or 12,000 values for every single parameter for each tumour. Now, there are two most recent papers at the bottom there by Katja Pinker that give a very comprehensive overview of really where we are with radiogenomics at the moment. So if you're interested, they're worth a read. So it's quite a tricky concept to get across, but essentially, radiomics is seeking to ask questions about the relationships between features that are seen in medical images and the biology of breast cancer. So we're talking about our standard radiology descriptors or BI-RADS morphological descriptors, but we're also talking about information which is unseen in the images, so the computer extracted quantitative data sets. And by combining these together with the histopathology and the genomics, it's hoped that there may be some sort of association or predictors that may be relevant to pertinent clinical questions. So to give an analogy, if you look at oncology practice, oncologists have many different predictive or prognostic models, things like Adjuvant Online or Predict. And into these models, they put a number of different parameters. things like patient age, tumour size, tumour grade, lymph node status. And then these models enable them to give information on long-term outcomes, so likelihood of effectiveness of chemotherapy, recurrence rates, metastatic disease. The analogy with radiogenomics and what radiogenomics is trying to do is create imaging phenotypes or imaging traits that in themselves may be able to predict on long-term outcomes. So if you say you've got one or a combination of imaging traits, that they might be able to, at outset, determine the likelihood of metastatic disease or the likelihood of recurrent disease. This is just another example of a radiogenomics approach from a radiology paper published in 2016, and you can see what people are trying to do: take the morphological aspects of a cancer, combine that with the quantitative data sets to look at potential associations with molecular subtypes and outcomes. And here are two quotes from that paper. Quantitative MR imaging radiomics shows promise as a means for image-based phenotyping in assessing the risk of cancer recurrence. And merging imaging phenotypes with genomic data in the future may lead to improved survival predictors. So there are many different terms used in this era of big data analysis and artificial intelligence. So just to clear up any confusion or misinterpretation, I think we'll go over a few definitions. The term omics refers to the totality of the object under study, so the study of large comprehensive biological data sets. The term genomics refers to the study of the genomes of living things, so looking at DNA sequencing and genetic mapping and putting this into large databases. Radiomics refers to the extraction of large amounts of quantitative features from medical images using data characterization algorithms. It's arisen from the field of oncology and is one of the most advanced applications in its field. It's essentially fast, rapid, high-throughput, reproducible extraction of quantitative imaging data sets, which are then transferred into mineable, usable data that may be able to be used to look for associations with specific features of interest or regions of interest. Now, the term radiogenomics in itself brings a little confusion because it's actually being used in two contexts. Historically, it's been used by radiation oncologists to describe the difference in sensitivity of a person to radiotherapy. So they've used it to describe an individual phenotype that may be associated with worse late toxicity to radiotherapy or improved outcomes to radiotherapy. However, in the context of imaging, and what we're talking about in this lecture, radiogenomics refers to the correlation between imaging features and gene expression. So the correlation of radiomics with genomics and the integration of quantitative imaging data with clinical features and outcomes. As I said, the vast majority of data at the moment analysis has been done using quantitative MR data, and the benefit of it is it gives representative whole tumour quantitative data sets. Now, we consider histopathology to be the gold standard, and it is in many genres, but we know when we're biopsying tumours at outset, we're only sampling part of the tumour. And the thinking behind it is if we've got large quantitative whole tumour, quantitative data sets, that we can move away from this selection bias that we have with core biopsy. So there are a whole host of functional MR sequences that we can use in this sort of analysis. And these are probably the more mainstream ones that we have available to us at the moment. We've got dynamic contrast enhanced MR, which gives information on vascularity, and diffusion weighted MR, which gives information on cellularity. And if we look at the data sets out there at the moment and the papers that are published, they've mostly employed the DCE and diffusion data that we've gathered over the last 10 to 20 years. But that doesn't mean that other functional sequences are excluded, and certainly in the future there is lots of potential for incorporation of spectroscopic data, R2 star data, and there's a whole host of preclinical quantitative functional imaging things like CEST imaging, carbon-13, which also would lend itself to this type of analysis in the hope that the sort of hidden features in the quantitative data sets may be able to provide information on long-term outcomes. We know from our standard time signal intensity curves that we can get a number of different semi-quantitative parameters, things like maximum signal intensity, relative signal intensity, initial area under the gadolinium curve, or enhancement fraction. And we have the facility also to do pharmacokinetic modeling, where we can derive parameters like K-trans, KEP, VE, and even perform textural analysis, all of which can be channeled into a radiogenomics approach. Diffusion-weighted imaging is probably less alien to a lot of us now, and certainly in the UK, many centres will incorporate a 2 to 3 minute diffusion sequence as part of their clinical protocol. And again, we can draw a region of interest around tumours on our ADC maps, and we can get ADC maps for every single voxel within that region of interest and sum it for the entire tumour. So this was essentially the landmark paper that got people very interested in radiogenomics. You can see immediately that we've got CT data here, so that shows you that this sort of approach is not exclusive to MR data. They use quantitative CT data. And this was a group in the Netherlands led by Ertz et al. And what they were able to do is they differentiated 5-year survival curves between non-small cell and small cell lung cancers using a non-invasive quantitative radiomics approach only. So the imaging features and the quantitative imaging features seen on CT were able to differentiate 5-year survival curves. So this was quite groundbreaking, and people got very interested and thought, is this going to work in my tumour group? And certainly it has been explored in other areas of radiology. There are a number of different websites, if you're interested, that have sprung up that have a lot more detail and information on the type of analysis that's been going on. Radiomics, Oncoradiomics, radiogenomics.org, if you want to look at them. And the fact that we're recognizing that certain types of breast cancer have specific imaging characteristics is not a new thing. You can see these papers published nearly a decade ago from Fiona Gilbert and Christiane Kuhl. We were already starting to recognize that certain breast cancers had quite specific imaging features. Because here we were looking at high-risk cancers, and we noticed that in BRCA1 carriers, the cancers were more likely to be smaller on MR than mammography, were more likely to be triple negative in molecular subtype, and were less likely to have microcalcification compared to cancers in BRCA2 carriers. And this is where we're moving on now from this with our radiogenomics approach, because we're able now to look at the large quantitative data sets that we've been able to obtain through our MR imaging over the last decade or so, in the hope that those features may be able to give some hidden information on long-term outcomes. So these are the different molecular subtypes of breast cancer, which people are looking really at initial associations with. These differ in their clinical features, their clinical courses, response to neoadjuvant chemotherapy, etc. And we determine these through immunohistochemical staining or our hormone receptor profile pattern. So we've got our luminal A and B cancers, which are the most prevalent with the best overall prognosis. The triple negative breast cancers, which are a smaller subgroup, have the worst overall prognosis, worse patterns of metastatic spread, and earlier disease relapse rates. And the HER2 positive cancers, which are somewhere in between with regards prognosis. And we know if we look at MRI now in assessing response through tumour subtype, that actually MR is more accurate when evaluating response in the triple negative breast cancers, the HER2 positive cancers and the high-grade tumours, but actually it has worse accuracy in the luminal cancers and low-grade tumours. And this is in part due to the way these cancers appear on imaging. Triple-negative breast cancers, although not across the board, and if you speak to any oncologist, they will tell you that there are probably at least 10 different subtypes of triple-negative breast cancer. Despite that, quite often they have a specific imaging phenotype, which is, as we see here, a well-defined rounded mass with pronounced peripheral enhancement and often high T2 weighting centrally. And again, that's often seen on ultrasound, where triple-negative breast cancers can mimic cysts. And you can see those features here are mirrored on the diffusion-weighted sequence with a very pronounced restricted diffusion rim and a free diffusion core. This is another example I'm grateful for Sarah from, looking at a triple-negative breast cancer where you can see it's a very pronounced, rounded mass with pronounced peripheral enhancement and a free diffusion core. And that matches frequently what we see on the histology, which is of central necrosis, and you can see the peripheral edge of this triple-negative breast cancer has a very smooth tumour edge. HER2-positive cancers, again, although not across the board, often are seen as rounded masses with homogeneous, strong enhancement. And you can imagine if your triple-negative and HER2-positive cancers are going to shrink down circumferentially, they'd be much easier to evaluate response accurately in, rather than if you've got a far more diffuse cancer. Or a luminal cancer. We know the luminal and low-grade cancers have often far more spiculation, as you can see on the T1 weighted image. You can see the enhancement in this cancer is less obvious, it's more homogeneous throughout compared to the triple-negative breast cancer, and the restricted diffusion on the diffusion weighted sequence is far more homogeneous throughout. And again, that's matched in this other example from Sarah. It does appear more like a rounded mass here, but again, there's spiculation, which helps distinguish it from a triple-negative breast cancer. And these features are matched with what we see in the histopathology. You can see that peripheral tumour edge is infiltrating into the fat, and it appears very different to the histology of a triple-negative breast cancer. So hopefully I've tried to illustrate a little bit about where we are with the radiogenomics approaches. We're looking at our MR data, pulling off the BI-RADS descriptors, the semi-automatic or automatically extracted quantitative data set, sometimes known as Haralick features, and linking that in the most simplistic terms with the molecular subtype of breast cancer, and if there are research groups out there with facility to do full genetic mapping with that sort of information. So where are we with publications? Well, there's a whole host of publications that I could show you with a lot of variety of results, all of which would probably be quite confusing. I've picked out a selection here. These on this slide really show a couple of groups that have looked at associations with molecular subtype. And you can see here, luminal B molecular subtype is associated with enhancement dynamics at MR imaging. Luminal A and B molecular subtypes associated with imaging features. So radiogenomics is really in its infancy now, and people are starting to explore any associations with their data. These two papers have gone a bit further. They obviously have the facility to do full gene expression mapping, full gene expression profiles, and you can see that they've looked for associations. And this slide really illustrates that also it's not confined to MR data. There are groups looking at quantitative digital mammographic data and any associations with molecular signatures. And in the paper at the bottom, there's a group that published MR imaging features are associated with distant metastasis-free survival. So starting to look at associations with longer-term outcomes. Just got a couple of slides looking at some of the, this is considered quite an important paper. It's published in 2012. This group were able to do global gene expression analysis on 10 preoperative MR patients. And they did high-level analysis looking at 21 imaging traits. And they found these were globally correlated with 71% of the total genes measured in breast cancer patients. So they found 12 imaging traits which were correlated with breast cancer gene sets and 11 imaging traits correlated with prognostic gene sets. And they went on in 2015 to investigate multi-scale relationships among quantitative computer vision extracted DCE MR phenotypes and look at associations with early metastasis and long coding RNA. And they found that 8 long coding RNA was associated with an enhancing rim fraction score, and that the enhancing rim fraction score was associated with early metastasis and a type of RNA which was a known predictor of poor metastasis survival. So what does it all mean, you may say? Well, exactly, what does it all mean? I think we're not entirely sure at the moment. People are starting to explore. Radiogenomics is in its infancy, and there's lots of potential in the future. There are a large number of advanced functional imaging biomarkers that we have available to us. As you've seen at the moment, the majority has been done using DC and diffusion data, but there's potential for lots more in the future. It does show great promise, though, to allow deeper insights to explore tumour biology by integrating the genomic and imaging data. We're far away now from, I think, developing this in clinical practice, and the role of radiogenomics in clinical practice needs establishing. And certainly, as with any of the quantitative imaging analysis, there needs standardization to validate any correlations. However, it's a very exciting and novel area of imaging. I expect to hear more about it in the next 10 years. Thank you very much for listening. I'm happy to take any questions.